So in this recording, we're going to investigate the semi-autonomous organelles that we find in eukaryotic cells. So all eukaryotic cells have mitochondria. So we can see here in our standard picture of an animal cell, these kind of kidney bean shaped, in this case sliced in half with a wiggly membrane on the inside, mitochondria. Now in plant cells, there's another semi-autonomous organelle that is called a chloroplast, or one of a many variety of plastids. But plant cells, as you can see, have mitochondria as well. So basically all eukaryotic organisms are going to have mitochondria, and those that photosynthesize are going to have chloroplasts, or members of the plastid family as well. Now, semi-autonomous means that they have some control over their own behavior. And the reason this is because they have some of their own DNA. So they're not part of the endomembrane system. They all have a double membrane, and that will come into play a little bit later why that's important. They have proteins made by free ribosomes, meaning they have their own sets of tiny ribosomes, and they contain their own DNA. This is why when we mentioned the nucleus, we've talked about most of the DNA in your cell is contained in the nucleus, but a very small fraction is actually found in the semi-autonomous organelles. Now, let's address mitochondria first and consider their role in the cell. Mitochondria are basically in charge of creating a pool of ATP. Now, ATP is an energy molecule that we will study in further detail later, but it is the form of chemical energy that biological systems use to power themselves. So mitochondria are the source of all the enzymes that are going to be used to convert your food basically into ATP. And so we can see this double membrane structure of the mitochondria, the outer membrane and then the inner membrane, and the space between would be the intermembrane space. Now the inner membrane is folded up upon itself to create what we call cristae, and that increases the surface area, giving you a larger surface area to put enzymes that are membrane bound into the inner membrane. And then of course the interior of the inner membrane we call the matrix. Now you can see here our cartoon version versus our transmission electron micrograph of the mitochondrion. Now most textbooks, most sources will show you mitochondria as these little kidney bean shaped organelles. But in fact, there's lots of data that suggests that mitochondria are very dynamic, that they divide and fuse with each other, and that in fact some cells you have a relatively dynamic reticulum of mitochondria. So remember, reticulum, like endoplasmic reticulum, means a network or net-like. And so we see here, using a special dye that just highlights mitochondria, you see not kidney bean shaped organelles, but rather a network of membranes that are energy producing. Now, the other semi-autonomous organelle that we're considering are the chloroplasts. Again, they're one member of a family of organelles called plastids. But the chloroplasts contain a green pigment called chlorophyll, and we'll discuss this in greater detail when we discuss photosynthesis. But this is going to be the site at which sunlight is captured and converted into a usable form of energy to fix carbon dioxide into sugars. Now, the chloroplast is a little bit more complicated in its structure. It in fact has three membranes, and the new membranes that we're going to add are thylakoid membranes, and there's going to be the stroma fluid in which those thylakoids float. So when we look at this three-dimensional model of the chloroplast, we can see we have an outer membrane and an inner membrane, in between which we have an intermembrane space. But inside the inner membrane, then, we have a separate network of membranes called the thylakoids, which form very specific structures called grana. And these are stacks, these like pancake stacks of membrane are going to be oriented in a way to capture sunlight very, very efficiently. And then the liquid inside that inner membrane in which the thylakoid floats is called the stroma. Again, we can look at our model, and in this model I like it because it shows you that the green pigment, the chlorophyll, is only contained within the thylakoid, while the outer and inner membrane, or the outer and inner envelopes of the chloroplast, are in fact relatively pigment free. Now again, you can compare our model to the transmission electron micrograph and see how we build from the actual micrographs into our models to understand these organelles.